clinical biochemistry. So that we need to understand the calibration too much deep way. Okay, and after completing today's class, hopefully you will not feel any kinds of doubt regarding calibration. See here, see. Calibration basically a procedure which ensuring that the laboratory instrument provide the accurate and reliable result. Accurate means the result should be 100% fully accurate, means right result. Okay, and reliable means that is dependable. Whatever result given by your instrument, that is dependable to the patient, so that patient can easily depend to your result, that is reliable. Okay, after that, the output signal with the known concentration of the analyte, so that there should be some known concentration of the analyte. We will uses these kinds of known concentration of analyte and understanding the mechanism of machines. Basically, it involves the solution with known concentration of analyte to teach the instrument how the measures accurately. Means whenever we are doing the calibration, it means that we are teaching to the instrument. This reagent is looking like that. This reagent will work like that. This test should be occurred like that. So that calibration is nothing but हमको सिखाना पड़ेगा. The instrument we are using, we have to teach to the instrument that what are the reagents mechanism as well as how to perform the test, so that we are using the calibrator to teaching the instrument. Okay, so that calibration is nothing but it is the procedure to teach the instrument to doing the test properly. Understood up to this much? Any doubt up to this much?
Yes, sir.
No, sir.
Understood.
Okay, see, now we are understanding the calibrator. Calibrator means use the calibrate use to means we have to use for calibrate the instrument. See here, calibrate the instrument and create a reference range. Getting my point? So that calibrator we are using to calibrate the instrument properly and to create a reference curve, so that we can easily measure the right result properly. Getting my point? So that calibrator is used to calibrate the instrument and create a reference curve for measuring the normal ranges. Okay. After that, standard, it is a pure substance. If I am saying about the standard of glucose, that is 99.9%, right? That is almost 100%. There will be no other particle should be present in in standard. Standard is only the pure substance. Okay, there should be present only the pure substance of this particular reagent. That is standard. After that, if I am coming to the control, see here, to ensuring the accuracy and reliability of the result to the routine testing means we have to uses control, means we have to uses the normal sample, normal sub good sample we are using to understanding the accuracy and reliability of the result in routine laboratory. Getting my point? So that first one is the calibrator, second one is the standard, and third one is the control. Okay, calibrator is using for calibrate the instrument to teach how to you run. Standard is a pure substance to taking the result of the test, and control we are using for maintaining the accuracy and reliability of this particular instrument. Okay, understood?
Yes,
Sir,
Yes,
sir.
sir.
repeat the point calibration.
Yes, Semi.
Sir, repeat the calibration point.
Calibrator. Calibration is different things. Calibration means you are teaching to the instrument how they will work into this particular reagent. Getting my point? That is calibration. And which kinds of analyte you are using for doing the calibration, that is called calibrated. Understood? Okay. Now, why calibration is required? See, firstly, patient safety. because without running the calibration, you will not get the proper result. Okay, how to run the calibration, what are the types, we will discuss it laterly. Okay, so that incorrect result if you are giving, then the wrong diagnosis might be found. Okay, if you are doing the incorrect result, then the wrong diagnosis should be found. If the wrong diagnosis should be found, then treatment should be wrong. Okay, first. Second thing is the productivity. Productivity defines that the ensuring the constant result across the run instrument and laboratories. Productivity means within the certain time, within the proper instrument, we are getting the proper result. That is called productivity. If you are not running the calibration properly, you will not get the productivity properly. Getting my point? That is productivity. After that, inter-laboratory comparison, standardized calibration ensures the uniformity of the test results. Means whenever you are running the calibration properly to the particular instrument, they will give you the good result. They will give you the proper result so that you can easily compare with the other laboratories, because your all test giving you the proper result. It will make up your confidence. Okay, so that it's called inter-laboratory comparison. Getting my point? After that, see here, complaints means calibration is required for adheration accreditation body like ISO, CAP, SLIA. These kinds of accreditation body require the calibration. They will check your machines did last calibrated, that they will check, as well as they will check the result of your calibration. I will teach you how to we are measuring these kinds of graph, as well as how to we are running the calibrator. I will teach you. Okay, I will teach you the whole theoretical part which is required for your practical laboratory. Okay, that's all are the importance of the calibration. Say me up to this much. Any doubt?
No, sir.
किसी का कोई दिक्कत है? कोई भी प्रॉब्लम नहीं है ना?
No, sir. But
बताओ बताओ अमित।
difference with the standard control, sir.
सुनाई नहीं दे रहा है बेटा, सुनाई नहीं दे रहा है। दिक्कत है, तुम्हारा नेटवर्क का
Actually,
दिक्कत है
sir, मैं
क्या?
एक्चुअली मैं ट्रैवलिंग कर रहा हूँ, बट मैं ट्रैवलिंग कर रहा हूँ, मैं सर क्लास अटेंड कर रहा हूँ। मेरे को डिफरेंस नहीं
कुछ
जानना है
भी
सर,
सुनाई नहीं दे रहा है,
स्टैंडर्ड।
कुछ भी सुनाई नहीं दे रहा है।
ओके
लेक्चर
ओके
खत्म होने के बाद
ओके सर।
क्वेश्चन करो। खत्म होने के बाद क्वेश्चन करो। ठीक
ओके
है,
ओके ओके ओके
ठीक है अमित। ठीक है, कुछ भी सुनाई नहीं दे रहा है, बहुत दिक्कत है। See, now we are understanding the types of calibration based on the purpose. See here, first types is the based on the purpose. See, primary calibration. What is primary calibration? I have to choose the another pen because rate is matching, so that we have to change the pen. Primary calibration use the primary standard, which is pure chemical compound with the known precious concentration. Getting my point? Primary calibration means firstly you have to uses the pure chemical concentration as well as the primary standard solution for running the primary calibration. Okay, like if I am giving you the example of glucose, that is 99.99% for the glucose acid, that is called pure glucose. So that we are saying that standard of glucose is 100%, right? Standard of glucose is 100%. Okay, so that is the primary calibration. Now I am coming to the secondary calibration. How it's look like? See, use the calibrator prepare from the primary standard, usually in the matrix that mimics the patient sample or serum. See, primary calibration is only the standard, but secondary calibration required. If that is the standard, there should be small amount of mimics, means into the surroundings there should be present patient serum or plasma. That is called secondary calibration. You can easily purchase it commercially because it commercially available. See here, serum based glucose calibrator. so that there should be glucose standard and there should be present the serum of some patient. Okay, that is the secondary calibration, because you are teaching to the instrument, so that you have to teach it's a serum, it's a reagent of glucose, it's the standard of glucose. Okay, so that it can easily commercially available, you can purchase it. That is secondary calibration. Primary calibration does not require any kinds of serum or plasma into their surroundings, but secondary calibration is required these kinds of things. Okay, clear? Based on the purpose, that is primary and secondary. See, based on the methodology, it is also important things about the calibration. You need to understand it, how it's look like, as well as which kinds of things are going on into this particular method. See, first one is the single point calibration. Here uses one calibrator with a single known concentration. Getting my point? Single point calibration means here required only one calibrator with known sample concentration or known signal concentration first. Secondly, simpler but less accurate. Too much simple and less accurate means that is accuracy level is low, because you are using it one single time, so that it's called single point calibration. Okay. After that example is zero calibrator for blanking. If you are measuring the blank, or if you are doing the blank into this particular instrument, then you might use the one calibrator, or that is single point calibrator. Okay. That's all about the single point calibration. Secondly, two-point calibration. Two-point calibration defines that one for zero baseline and another one for the higher concentration of standard. Getting my point? If you are using only one calibrator, that is called single point calibration. If you are using two calibrator, one for zero baseline and another one for the high standard concentration, that is called two-point calibration. If I am giving you one example, that for glucose calibrator at 0 milligram per deciliter, means you are using the calibrator in zero baseline and you are using the calibrator and 100 milligram per deciliter, because that is a high standard amount, right? We are using for zero baseline as well as we are using for the high standard baseline as well as high standard concentration. Zero for the zero concentration is for zero baseline and 100 concentration for the high standard concentration. Getting my point? That is two-point calibration. Understood? Two-point. And nextly I am explaining the multi-point, then you will say. Okay. After that, multi-point calibration here basically uses several calibrators. Getting my point? Not only zero, but also another types of calibrator we are using here. Okay, uses several calibrator. Example, 5 or more. That might be 5 or that might be more than 5. Okay, across the range of this concentration, that is started from zero and concentration should be increases. Getting my point? Suitable for the assay with non-linear relationship. Non-linear relationship means whenever I will teach you the enzyme kinetic, then we will understand. Non-linear relationship means when you are doing any kinds of kinetic procedure like SGPT, SGOT, then within the interval you have to measure the OD, right? That is OD. Del OD defines that interval within this time and this time interval you are getting the OD. So that here the graph is not linear, right? If I am saying that graph is linear, then the graph should be like that. If I am saying the graph is not linear, then the graph should be like that. Okay, that is not linear. So that into the enzymatic assay, that should be not linear, as well as that should be non-linear. Here we are using the multi-point calibration from 0 to 100 and as well as 0 to various type of more than 5 calibrator we are using. So that if I am summing these things, that the methodology there should be single point calibration, double point calibration or dual point two point calibration, as well as last one is the multi-point calibration. Understood? Up to this much
Yes,
clear?
sir.
Okay, up to this much clear? Up to this much? Any doubt?
No, sir.
Okay, fine. So that we have understood the types of calibration, right? See, so that we have already understood what is calibration. We have understood what are the types of calibration. We have already understood what are the types that is based on methodology, based on the purpose of calibration. Okay. Now we are coming to the materials used in calibration. Now we have to understand that what are the materials basically uses for calibration. Okay, we need to understand that. Here basically I have already mentioned you that there should be calibrator you have to uses, standard you have to uses, and control or quality control you have to uses. So that we might uses three calibrator, right? We might uses three types of things for a calibration. That should be calibrator, that should be standard, and that should be control. See, calibrator is basically the solution with is preciously known concentration. Getting my point? Calibrator you can easily purchase commercially as well. Okay, there should be known concentration of the particular solution as well as must be traceable to the international standard. Okay, you have to match the international organization as well as standard, uh, international standard to the calibrator. See, that is NIST, who they all are the standard they all are the standard organization as well as international organization. After that, matrix matched to the mimics patient sample like serum and plasma. Getting my point? So the calibrator, I have already told you that calibrator is a known concentration of the solution. There might be present the matrix mesh that might be serum or plasma. That is calibrator. We can use the calibrator for calibration. After that, we can use the standard high purity of the substance. There should not be any other substance. There should be present only pure this substance which standard we are using. That might be sugar, that might be urea, that might be any other substance. In which substance we are using, we have to uses only this substance as a standard. Getting my point? And the example is 99 to 99.99% of pure glucose. Okay, that is the standard we have understood. Now we are coming to the quality control sample. No directly used for calibration but verify its accuracy. They basically not uses directly for the calibration procedure. Okay, but sometimes we are verifying the accuracy of this particular quality control serum with low, high, and normal high concentration. They all are the quality control sample. Keep in mind that whenever we are running the control, that is look like normal sample. Okay, control means we have to choose in some healthy sample. We have to running these kinds of sample, that is called control sample, as well as that is called quality control sample. Getting my point? So that we have understood what are the measures as well as what are the requirements of quality control, calibration. First one is the semi, first one is first one is calibrator, and second one should be sample, and third one should be sample means quality control sample, and third one should be standard, right? These three quality control and these three calibration materials is used for calibration. Say me up to this much, any doubt?
No, sir.
Okay, hopefully you have understood the theme. Okay, that's all are the material used in calibration. किसी का कोई दिक्कत है अप टू दिस मच? कोई दिक्कत तो नहीं है? बताओ बताओ बताओ, कोई प्रॉब्लम है? ओके, आई होप यू ऑल आर
हेलो
अंडरस्टैंड.
सर।
हेलो गौर.
আমি বলছি স্যার ক্যালিব্রেশনের মূল কাজটা কি?
মেশিনটাকে শেখানো
ठीक है सर। ओके ओके।
how to teach, how to run. Without running the calibration, your machine will not learn how to do the action.
ठीक है सर।
Okay,
ओके।
okay, okay. See, now we are coming to the workflow, how it's happening. Okay, how the calibration will happening, we need to understand it. Okay, see, calibration workflow in our biochemistry laboratory. Okay, first one is the step one is preparation. Preparation means firstly you have to taking some preparation. Second step is you have to verification, you have to verify it. Sorry, firstly preparation, secondly you have to run the calibration, thirdly you have to verify it, after that you have to generate the calibration curve, after that that is the example. Okay, okay, see here, now I am discussing with you how it's happening. See, firstly preparation, choose the appropriate calibrator. Firstly you have to choose the appropriate calibrator. That's depend upon which kinds of reaction should be happening. The reaction that might be enzymatic, that might be colorimetric, that might be semi-auto, that might be automatic, that might be immunoassay. So that you have to choose the calibrator based upon the reaction is happening. Okay. Secondly, you have to sir tell me the teammates के बोलिए ना कोई क्वेरी है तो क्या बताओ बताओ किसी का कोई क्वेरी है बांग्ला या अदर लैंग्वेज में करते हैं तो नहीं 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 बेटा नहीं बेटा देयर इज ओनली वन क्वेश्चंस ओके 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 देयर इज ओनली वन क्वेश्चन आई एम गिविंग द आंसर इन इंग्लिश ओके and the previous one question is what is the main function of calibration. I have given the answer. Understand? There have also various type of students are present from Hindi, Bengali, English. Okay, so that anyone can ask the regional language, but I will give you the answer in English. Okay, okay. कोई बात नहीं बेटा हो जाएगा सब कुछ हो जाएगा लैंग्वेज की तो बात है okay 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 so here so that if anyone are asking any kinds of question their regional language firstly I will repeat the question in English. Okay, after that I will give the answer, don't worry. Okay, see, now we are coming to the preparation section. How do we prepare the calibration? How do we prepare? See, use the appropriate calibrator. I have already told you that that's depend upon the reaction. Which kinds of reaction might be? That might be enzymatic, that might be colorimetric, that might be immunoassay. Okay, and you have to choose in the calibrator before expiration date. But if the date is expired, then that will be not work. Okay, so that you have to choose the calibrator properly within the expiration date. Understood? Secondly, that is instrument setup. You have to clean the instrument properly. You have to check the instrument are functioning properly or not, as well as you have to count how much number of reagent is already present into the particular instrument, as well as how the control is running, as well as you have to check the reagent expiration date, as well as whenever you are doing the work into any kinds of laboratory you might see that there should be lot number of the instrument, lot number of the reagent, right? You have to match with the lot number with the particular date. If it's expired then you should not use it. Okay, so that firstly you have to choose the calibrator, after that you have to choose the instrument and set up the instrument properly. Hardly if any kinds of standard you are using as as an calibrator, then you have to prepare it. If you are preparing it by oneself, then prepare it, or you can purchase it commercially. Getting my point? See, if any kinds of calibration require the standard, then you have to choose the standard, as well as you can prepare it into the laboratory, or you can purchase it the standard for running the calibration. Okay, understood? That's all at the preparation. You have to take the preparation at first. Okay? Are you prepared? Understood? Up to this much? Up to this much? Understood?
Yes,
Anyone?
sir.
Anyone have any problem? ठीक है. I think that everyone are understanding. किसी का कोई दिक्कत नहीं है? Okay, okay, बेटा. Okay, I'm moving. Okay. Now I am coming to the calibration. So that we have already understood the preparation section. Now I am coming to the running of calibration or running the calibration. We have to run the calibration. Firstly, we have prepared. Secondly, we have to running the calibration. It's not possible to seeing these kinds of things into machines because every laboratory have their own machines. Every laboratory have their own taste. So that whenever you will go to the laboratory, you might do these kinds of practical things as well. See, load the calibrator onto the instrument from low to higher concentration so that we have understood, right? There might be single level, a single point calibration, dual point calibration, multi-point calibration, right? If it's multi-point calibration, then you have to choose in the low concentration at first. After that you have to move gradually to the higher concentration. Getting my point? That is the multi-point calibration, as well as that is the load of calibrator. But if it's required only one, then you have to use this one calibrator to load the instrument properly, as well as you have to maintain the protocol of this particular instrument properly. Okay, so that you have already run the calibration or calibrator you have already run. Second, signal measurement. You have to measure the signal like absorbance. You have to measure. If we are doing any kinds of fluorescent test, then we have to understand the fluorescence. If we are doing any kinds of immunoassay, then we have to understood the immunosense for each calorie calibrator. Okay, firstly you have to run the calibrator. Secondly, you have to measure the signal, how they are giving to the signal. Otherwise, the graph will not form. Okay, they are collecting the data of the particular calibrator. After that, generate the calibration curve. Plot the instrument signal or absorbance on y-axis against to the calibrator known concentration in x-axis, so that calibrator have their own some results. Okay, so that if I am doing one thing, that I will explain the whole things, don't worry. If I am doing one thing, that is the graph, that is the y-axis, that is the x-axis, right? So that you have to present the known concentration of this particular calibrator here, and you have to put here the concentration you are getting from the instrument. Okay, then x and y axis graph should be formed. Okay, and the data should be in a linear form, means like that, or non-linear form like that. Getting my point? After that, calibration graph should be formed, that is predefined statistical r square more than 99 for linearity. If it's linear, then r square should be higher than 0.99. If it's lower than 0.99, then your calibration is not done properly. Then you have to run the calibrator, or you have to learn the calibration properly again. Okay, so that if I'm sum upping these things, partly you have to load the calibrator. After that, you have to measure the signal of this particular calibrator. After that, you have to generate the graph. After that, you have to compare the graph with this particular normal concentration. Getting my point? Up to this much, and we will understand it how it's happening. Up to this much, any problem?
No, sir.
No problem. Okay, anyone facing any kinds of problem up to this much? Anyone? I think no, right? Mashina, are you understanding?
Yes, sir.
Okay, fine. Now it is the high time to discuss about the verification thing. So that firstly we have to prepare the things here. We have to prepare. After that we have to run. After that we have to verify it. After that we will see the calibration graph here. Verification. Run the quality control. See, uses the QC sample with low, normal, and high concentration to verify the calibration, so that you have to run the quality control after doing the calibration, or after running the calibrator you have to run the quality sample. Otherwise you will not able to understand that is the machine is giving you proper result or not. So that if I am giving you one example, that whenever we are teaching the student, after teaching the student we are taking the examination, right? Whenever we are taking the examination we are providing the questions, right? Whatever question they have already understood, whatever concept they are already understood, after that we are taking the test, right, for checking their knowledge. That is also like that. Whatever teaching we have already done to the instrument, and whatever calibrator we have already learned, we have already learned, we have to check, we have to imagine, as well as we have to taking the examination of the machines by running the quality control. So that whenever we are running the quality control we will get the result. There will be the quality control. There will be first one is the low, after the normal, after the high concentration, to verify the calibration, is it proper or is it not. If we are running the normal firstly, after that high, after that low, okay, so that we can easily imagine that is the machines is running properly or not. Getting my point? And result should be for fall within the acceptable range. So that if result are we are getting the result within the acceptable range, then we have to accept it. Otherwise we have to deny it. Okay, see, if if you see or quality control result are out of range, until or unless you are getting the proper result of from this particular instrument, from this particular machine, you have to run it, you have to recalibrate it, until or unless they are getting the proper result or proper value or proper reference range of this particular reagent of this particular sample, we have to run it. Okay, if it is out of standard range, then we have to running the calibration again. Getting my point? That is the verification. If I am sum upping these things, first one is the preparation, second one is the running the calibrator, and third one is the verification stage. Into the preparation stage we have to prepare the calibrator, we have to prepare the machines, right? As well as if standard we are using, we have to prepare the standard. After that, in the running stage, firstly we have to running the calibrator. After that we have to getting the measurement of the signals. After that we have to make the graph. After that we have graph means make the curve. After that we have to check the curve properly, is it linear or non-linear. In the verification section we have to use some quality control sample, so that we can easily verify the things, is it proper or not. Okay, up to this much any doubt? Up to this much any doubt?
No, sir.
So if I understood.
Yeah, yes, sir.
Okay, anyone facing any doubt? Anyone?
No, sir.
Okay, understood. Okay. Okay. Now, see, trouble short potential might be happen. There should be reagent degradation, pipetting, error. So that we have understood, right? Into the quality control, we have understood the internal quality control, yes or no? Internal quality
Yes, sir.
control, and another one is the external quality control, right? So that can anyone say me pipetting error is it? It is in which type of error?
Internal
Internal,
error, sir.
right?
Analytical error.
Very good. There was analytical error. Are you understanding the things? Pre-analytical, analytical, and post-analytical error. Yes or no? Into the analytical there was
Yes.
random and systematic. Payal, can you say repeating error is random or systematic?
Random.
Random. Very good. Very good. Very good. So that we have understood like that. right? What are the types of error? Analytical, pre-analytical, and post-analytical. Can anyone say me which is under pre-analytical? One example, just give me one example of pre-analytical error. Pre-analytical error, one example give me. Wrong sample collection, right? And within the analytical, say me one example of analytical error. Instrumentation problem, instrument fault, instrument failure, right? And can anyone say me what are the post-analytical error? What is under post-analytical error? Can anyone? Post-analytical error? Like documentation, right? And report generating, yes or no? Topography, understood. And these kinds of error we have already understood. Okay, up to this much, anyone have any doubt? Up to this much, so that up to this much, whatever we have understood, up to this much we have understood what is calibrated, what is standard, what is control. Anyone have any questions regarding these things? Anyone have any doubt? After that, we have understood what are the types of calibration, right? One is based on the purpose, another one is based on the sources. Into the purpose, there is primary and secondary. Into the instrumentation or into the signal measurement, there is a three types. One is single, another one is a dual, and another one is a multi, right? Single point calibration, dual point calibration, and multi point calibration. Understood? After that, we have understood what are the process of calibration. Firstly, preparation, right? Preparation. And secondly, running of the calibration. Running of the calibration. And thirdly, semi. Third one, semi. Verification. Very good, very good, very good. Verification. Anyone have any doubt? Are you understanding the things properly? What's basically going on? Understood? Okay. See here. See here. Now we are analysis the curve. Please focus here properly. If you are not able to understand, say me. One time not, 100 times. Because I am here to teaching you all. To giving the learning mates to all. See here. Calibration curve. We have to understand the calibration curve, right? See. So that here is the x-axis and here is the y-axis. Yes or no? X-axis and y-axis. See, y is equal to signal, y is equal to m plus x plus c. See here, m is equal to slope. Slope means like that. Slope means like that. Getting my point? M is equal to slope. X is equal to concentration. Concentration. This one is a concentration. Yes or no? Concentration. Concentration. And c is equal to intercept within 2. Within 2, what are the intercepts? Getting my point? That is also r square. So that r square is equal to, we can say y, right? And y is equal to mx plus c. Understood? Understood?
Yes, sir.
Understood? See here, that is the known concentration. That is the known concentration 224. That is the known concentration. Okay, and that is the absorbance whatever we are getting from the machines. Yes or no? Instrument whatever we are getting. The first 0.02, 0.04, 0.06, 0.08, whenever we are getting, we have just put it here, right? We have just put it here and we have to match with here. That is the slope, right? And we have to just put it here, here. And whenever we are getting these kinds of line, so that we are getting the graph that is linear graph, right? Is there any kinds of helical? Is there any kinds of spherical? Is there any kinds of round? Is there any kinds of another shape? No, that is linear graph we are getting. So that I have already told you that if the value less than 99, if the value less than 99, if the r square is the value, r square less than 99, we cannot taking this result. See here. See, getting my point? R square should be always 0.99. Okay, see here. Here is the R square is equal to 0.9993. So that r square should be always 0.99. So that here is an higher than 99. Yes or no? Is it higher or lower than 0.99? Higher, right? This value is higher. And how can we get this value? This value we are getting from y, right? And whenever that is higher than 0.99, see here, higher than 0.99, then we can take the result. Then we can say that the graph is too much right. As well as we can checking this graph for the calibration. Understood? Any doubt? If it's less than 0.99, then we cannot take the result. Okay? Understood?
Yes, sir.
Okay. So that this graph is understood, that is linear graph, right? Because there is a no curve shape. It is linear, right? It is the linear graph or linear curve. See, non-linear curve. Is it linear or non-linear? Semi. Is it linear or non-linear? Semi.
Non-linear.
Non-linear, right? Because that is not a straight line. If it's a straight line, then we can say that it's linear, but it is not a straight line, right? That is a sigmoid-like shape. That is a sigmoid-like shape. See, immunoassay required 4 to 5 parameter or 4 or 5 parameter in logistic bit. Getting my point? Immunoassay basically required lots of parameter. Here, basically, you will get the non-linear curve. That is the known concentration, and this kinds of concentration we are getting by the instrument. See, 0, for 20, 40, 60, and these kinds of things we are getting from the instrument, and we have to write down the curve here. Here is also you can get that the r square is higher than, see here, r square is, see here, higher than 0.9974. So that r square is 0.9974. Is it higher or lower? Higher, so that we can check the value of the calibration curve. Understood? And how the r square you are calculating, I have already told you that. Understood? If it's less than 0.99, if it's less than 0.99, you cannot take the result. Then you have to run the calibration again. Understood? Any doubt?
No, sir.
Anyone have any doubt? No. See here. Now we are just taking one example. Okay, we are taking one example. Glucose we have to test in the spectrophotometry. I have already told you that we have to run the calibration in various procedures. That might be enzymatic, that might be immunoassay, that might be spectrophotometric, right? So that's it. First of all, what will you do? Example, reagent. So that in which procedure glucose tests are happening? Can anyone say? Which procedure we are testing the glucose? Can anyone say?
Enzymatic method.
Enzymatic method. Which enzymatic method? Can you say me the
God
method?
pot.
Godpot. Very good. Very good. Godpot, right? What is the full form of Godpot?
Glucose oxidase
Glucose
peroxidase.
oxidase and peroxidase.
Very good. Which enzyme basically uses here? And which enzyme activity? Hexokinase, yes or no?
Yes.
Yes.
Hexokinase.
Yes, yes. Bahar, how are you? Many days later.
Fine.
Fine. In the Murshidabad, there is a various type of problems that are occurring. I am seeing into the news. Are you fine? Into this critical condition?
Fine.
Okay, okay. Just take care of yourself. Just save from these kinds of, what can I say? These kinds of problem. Okay, fine. Okay. So that if I'm saying you one more questions for doing the glucose test, in which while we are taking the blood,
Gray.
that is the,
Chloride vial.
yeah. Can anyone say me without the sodium fluoride here is also another substance is used? Which substance is there? Which substance is there?
Sodium oxalate.
Very good. Why we are using sodium oxalate and why you are using sodium fluoride? Machina, why we are using sodium fluoride? Anyone? Sodium fluoride is using for stopping the activity of hexokinase, right? And sodium and potassium oxalate we are using for anticoagulant. Getting my point? Don't forget these things. Okay, see. See, so that glucose oxidase and peroxidase is the reagent. Here we are taking, it is an example. Not we are taking this kinds of calibrator all time. That is example. See, we are taking the calibrator that is, so that calibration or calibrator is known concentration. Yes or no? Known concentration we are using. That is 0 milligram per deciliter we are taking 50, 100, 200 and 400. Here we are using multi-level calibrator. Okay, so that procedure measure the blank with 0 mg per dl calibrator. Okay, after that measures the absorbance for each calibrator. After that the calibration graph will be generated. After that valid with the curve with QC sample. So that firstly you have to use as 0. After that you have to use as 50. After that you have to use as the 100. After that you have to use as 200. After that you have to use as 400. And right now nowadays machines will give you the calibration graph. Yes or no? Machine will give you the graph of calibration. What will you do? You have to match with the quality control sample. After that, that is the calibration section is done. After that you have to run the control as well as quality control sample. Then you have also got this kinds of graph or this kinds of card just matched with them. If the R value is higher than 0.99, then we can take the calibration, otherwise we have to recalibrate it. Getting my point? So that firstly you have to getting the calibrator curve. After that you have to getting the quality control curve. After that you have to match with this as well as compare with this, so that you are getting the proper value. Getting my point? If the R square is higher than 0, then we can take the blood. Okay, patient sample you can now measure. Okay, so that firstly you have to choose in the calibrator. It's too much easy. After that you have to measure as well as you have to use as the calibrator one by one. After that the graph will be appear. If the carb's value is less than higher than R 000 higher than 0.99 R square value, then we can use as the machines as well as we can use as the patient sample as well as we can measure the patient sample. Getting my point? Understood everyone?
Yes, sir.
Okay, anyone have any doubt? I think no. See, and common card here is also some same kinds of card. See here, same kinds of card and same kinds of error might be happening. Pre-analytical, incorrect, standard or calibrator. Okay, use expired reagent if you are using then pre-analytical error. Getting my point? Because the analysis is not happening right now. See, now analytical curve, if any kinds of faultation or if any kinds of failure of instrument, that is the analytical curve. Contamination of sample, if the sample is contaminated, then analytical error might be happen, so that we will not get the calibration properly, right? Post analytical incorrect intercept of the calibration curve, if the curve is not generated properly, that is the non-linear and that is the linear. Okay, that is non-linear, that is not linear. And failure to validate the QC, if you are not getting the calibration curve properly, you will not match with the QC sample, right? So that if I am sum upping these things, P analytical means standard or calibrator is wrong or expired region you are using. Analytical means there should be any kinds of instrumentation fault, and post analytical should be there should be the curve of calibration as well as incorrect. Okay, as well as there is a no validation of QC. Getting my point? Understood up to this much? Up to this much? Understood? Semi, up to this much understood?
Yes, sir.
Okay, fine.
Yes, sir.
That is a troubleshooting issues. Troubleshooting means how do you troubleshoot is QC result out of range. If any kinds of result or QC result is outshooting as well as out of range, then you have to fix the machines properly. If any kind of non-linearity of the curve, if the curve and the desirable curve is linear but the curve are getting non-linear, so that you have to troubleshoot or you have to fix it. If any kinds of instrumentation defect is problem, then you have to maintain or you have to fix the instrument properly. See, here is the basically the difference. It's too much important. What is calibrator, what is standard, what is control? So the calibrator for set up the instrument, standard, it is a pure substance for measuring the test, as well as control for verifying the accuracy and precision. Understood? Purpose to establish the relationship between the signal as well as concentration and analytical concentration. Signal and analytical concentration means that is the OD, right? That is the unknown value and that is the known value. Yes or no? That is the known value. So that it basically doing the relation, it basically creating the relation between the unknown concentration with known concentration. Understood? See, after that, providing the reference range, standard will give you the reference range. Control will ensure that your instrument is proper. Okay, metrics here should be present, some kinds of metrics, because that is calibrated. Here should not be any kinds of metrics. Getting my point? Because that is only pure. And if I am control, there should not also any kinds of metrics. There should be realistic patient sample you have to use as control. Understood? Metric should be present only calibrated, and there should not be any kinds of metric should be present in standard. There should not be any kinds of metric present in the control, because control we are running as the patient sample, that as in control. Okay, uses for the calibration procedure, standard for easily getting any kinds of routine laboratory or for easily getting any kinds of result, and control uses in daily basis for check the result. Okay, example, calibrator is glucose calibrator is 100 milligram per deciliter and standard is 99.99%, and serum control we are using for a low, normal and higher. Up to this much, any doubt? I have already explained the whole things regarding calibration, as well as I have already told you the extra things, that is what are the difference between the calibrator, standard and control, so that you can getting the basic idea of these things, as well as you can easily differentiate what's the things going on. Okay, so that I have mentioned here the differentiation. Say me, up to this much, any doubt?
No, sir.
Anyone facing any kinds of doubt up to this much? Anyone have any doubt? Anyone facing any doubt? Anyone have any
No,
question?
sir.
Okay. Payal, how have you spent your new year? Was it fine?
I was at home.
Understood. You have learned lot. You have done lots of activity, right? Because you are from West Bengal, so that I am talking with you. Okay. Anyone have any questions? Any doubt? किसी का कोई दिक्कत है? कोई क्वेश्चन है? कुछ भी नहीं है? Any kinds of questions? Is it too much tough? Is it too much tough? Calibration too much tough? Concept? No, it's really too much easy concept. You have to just memorize the things. You have to just look down upon the things whenever I will provide you into your group. Okay. Okay. Anyone wanna say anything? Anyone? Okay. I think that everyone understood. So that thanks a lot for joining today's class up to this much. Hopefully we will meet in tomorrow also, because that is yesterday's class. Okay, and we will meet tomorrow with another new topic, that's in point and kinetic. That will be also interesting for you all, guys. Okay, we will meet in tomorrow's class. Okay, today's class up to this much. Thanks a lot for joining.